All right, hello, and welcome to our third episode of our series highlighting current and exciting research projects in the MGNet Research Consortium. My name is Anusha Khan, and I am the project manager at MGNet Research Consortium. I will be your host for today. I'm joined by Dr. Patricia Sikorski, a researcher at George Washington University. I'm also joined by Ms. Rachel Canteri, our MG patient advocate. So in this episode, we will be featuring Dr. Sikorski's project titled Atypical B cells in Myasthenia Gravis. I now will give you both the floor to discuss this important topic in research, MG research.
Hello, everyone. First of all, I want to thank the MGNet for the opportunity to share my research and, of course, the patients for their participation. And Rachel, thank you for being here today. It's such a pleasure to have this discussion with you.
Yeah, thank you for having me. So I guess we'll go ahead and get started. I love to see that there is continued research done on this topic, just considering how rare the disease is and how much we still need to know about it. So to kick us off, our first question is, what is your research about?
Sure. So my research focuses on a specific type of cell in our immune system called B cells. And as some of you might know, B cells make antibodies that keep us safe from being sick. But in people with myasthenia, something goes wrong. So these B cells, some of them start making antibodies that attack our own proteins that are important for muscle and nerve communication. And this attack is what causes the muscle weakness. And there are two types of harmful antibodies in MG that are made. So those against the acetylcholine receptor, or AChR-MG, and antibodies against muscle-specific kinase, or MuSK. We know these antibodies are harmful and they cause the disease, but we really don't understand what causes the B cells to make these harmful antibodies. So my research project is about a unique type of B cell called atypical B cells. And these cells, as their name suggests, are atypical because they have unusual features that sets them apart from other types of B cells or normal B cells. So why are these cells important? Well, there's some indication that atypical B cells are involved in autoimmune diseases because they're found in higher numbers than normal in the blood of patients with autoimmune disease. And sometimes they can make these harmful antibodies. So it could be a really good idea to find ways to stop these cells from causing trouble. So no one's really studied these cells in myasthenia gravis. So the goal of my project was to characterize atypical B cells in both acetylcholine receptor positive MG and MuSK positive MG, and then compare them to people without autoimmune disease. And really briefly, the results of my study showed that there were higher levels of these atypical B cells in the blood of MG patients. But what I found most surprising was that I found different types of atypical B cells that were associated with each MG subtype. So the atypical B cells in the MuSK patients were very different from those in the AChR-MG, both in the appearance as well as some of the gene activity inside of these cells. So it really shows that something different is going on in these two MG subtypes. And then lastly, when we grew these cells in the lab, we found that they make a lot more antibody than normal B cells, and that some of these antibodies are harmful antibodies.
That's
Those are the
super
main
interesting.
findings. Yeah.
That's very interesting. So why is this research important, and where will you take it in the future?
Sure. So my study is important because it shows that these atypical B cells might be involved in MG, and that suggests they could be a good therapeutic target. I also found that there are these differences between these two MG subtypes, and that's important because it reveals that AChR-MG and MuSK-MG are distinct immunological diseases, suggesting that there are different environments that can lead to different cellular responses and disease development. Where this is going, so I'm continuing to study these cells. So right now I'm growing them in the lab to study these behaviors. And since I found that they do produce these harmful antibodies, I now want to know how I can stop them from making these antibodies. So to do that, I'm looking at the gene activity inside these cells to help us figure out what really makes them tick. And if we know what genes cause this behavior, we can design drugs to specifically target them.
That's super exciting. I know I mentioned to you that I work in a different field of medical as a vendor, but I speak some of this talk, so it's very exciting to see that something like this could be on the horizon for patients like myself. So the next question is, what is the most important insight you have for me or others as patients, and how will this impact us that do have an MG diagnosis, whether it be acetylcholine or MuSK?
Sure. So I think this research can give us some new insights into the immune system of patients with MG, and that can help us figure out better treatments. So right now, the treatments for MG aren't very specific, and they can cause a lot of unwanted side effects. So what we really need are more precise therapies for MG. And I think this step is one small step closer towards that goal by studying these cells and understanding them. So we really, rather than suppressing an entire immune system or getting rid of all of the B cells, we want to be able to target just the harmful ones before they start producing bad antibodies, and then save the good ones so that they can protect us. I guess also in terms of the differences that we're seeing between AChR and MuSK-MG, this is really biologically important. If we think about this in terms of trying to find new treatments, treatments are designed to target specific mechanisms, and a treatment that works well for one subtype might not be effective for another subtype of the disease, like AChR and MuSK. And that's because if it doesn't, it won't work because it's not targeting the right or relevant biological pathway. So by starting to understand these biological differences between these MG subtypes, I think that is really the key for developing these more effective and personalized treatment strategies.
Thank you for that response. I know that having personally gone through this, I'm relating to a lot of this information quite a bit. I'm sure others who will watch this will as well. So there's only one more question here, and it is, you mentioned that the B cells, the atypical B cells, were different in the ACH and MuSK patients. So what are some precipitating factors that might activate these B cells into behaving in this manner in both types?
That's a very good question. So that's what we're kind of delving into right now. And I think by looking at the genetic or the gene activity inside these cells, we can kind of figure out if there are different signals that are activating them in different ways and causing them to develop differently. So I think that's going to be hopefully in the future give you a better answer for that question. But yeah.
Well, thank you so much for taking the time to answer a few questions for all of us out here. And thank you for the research that you're doing in order to help make us healthier as a patient population.
You're welcome.
Thank you both so much for your time, for, conducting this important discussion. I think these discussions are super important in the MG space, and it's going to be super helpful for these patients and their families. We hope this information will be a great resource for the MG community. Thank you both so much for your time.
Thank you.
Thank you so much.